A+ലേക്കുള്ള വഴി ഇനി തനിയെ തുറക്കാം. Subscribe to the ENCLAPS channel. Ring the bell button and open success. It's that easy.
Welcome to the wonders of the biological world. Genetic engineering has progressed a lot more from biotechnology. Today, genetic modification in organisms can be implemented more effectively. This is made possible through the insertion of genes that code for desirable characters into the genetic constitution of an organism. Proteins that can be used for the treatment of diseases in humans are produced through genetic engineering. Now let's see a video based on a table. Proteins used in treatment, diseases. Interferons are the proteins required for the treatment of viral diseases. Insulin is used for the treatment of diabetes. Endorphin is a protein required for the symptom pain. Somatotropin is a protein required for the growth disorders. One of the future promises of genetic engineering is farm animals. The genes responsible for the production of insulin and growth hormones required for humans are inserted into animals like cows, pigs, etc., transforming them into farm animals. But there are certain limitations in producing insulin using bacteria. The most important hurdle in this field is the culturing of bacteria. The procedure is very sensitive and requires a lot of patience. Researchers in the field showed that instead of this, medicines can be extracted from the blood or milk of genetically modified animals. Genetic modification is implemented not only in animals but also in plants also. Today, insect-resistant plants like BT brinjal, soybean, BT cotton, maize, etc. are very common. When genetic modification is carried out in organisms, it should be assured that there is no harmful consequences to human or nature. Did you notice the headline of the newspaper report? How are the persons identified through DNA testing? The technology used for testing or the arrangement of nucleotides is called DNA profiling. Certain experiments was conducted by a scientist named Alex Jeffrey in 1984, paved the way for DNA testing. Just like the differences in the fingerprint of each person, the arrangement of nucleotides in each person also differs. This discovery became the basis of DNA testing. Hence, the technology is called DNA fingerprinting. Now, let's understand what the technology really means. DNA fingerprinting. The arrangement of nucleotides among close relatives has many similarities. Hence, DNA fingerprinting is helpful to find out the hereditary characteristics to identify the real parents in case of parental dispute and to identify persons found after long periods of missing due to natural calamities or war. In a crime scene also, DNA from the skin, hair, nail, blood and other body fluids obtained at the place of murder, robbery, etc. are used to compare with the DNA of the suspected person. Thus, the real culprit can be identified from among the suspected persons through this method. Now see if you can explain these indicators. Basis of the technology of DNA fingerprinting. The arrangement of nucleotides in the DNA of each person differs. This finding is the basis of DNA testing. Scope of DNA fingerprinting. DNA fingerprinting is helpful to find out the hereditary characteristics to identify the real parents in case of parental dispute and to identify the persons found after long period of missing due to natural calamities. Again, in a crime scene, the DNA from the blood, skin, nail and other body fluids obtained at the place of murder or robbery can be compared with the DNA of the suspected persons. Thus, the real culprit can be identified from among the suspected persons through this method. Now observe the collage given below. It is criticized that genetically modified varieties are a threat to the indigenous varieties and may cause health issues to the humans. There are possibilities to use the genetic modified organisms as bioweapons that can be applied to any country to their enemies. Such a situation is called biowar. This becomes a threat to the existence of human beings. So just like a coin, every discovery has a positive and a negative side. But remember, negative side arises only due to bad intentions. Now, the last question. Is it right to misuse the technologies that can pave the way to human progress? As such possibilities prevail, must we promote genetic engineering? No. Though there are certain possibilities of misuse, genetic technology has many merits, like making medicines, vaccines, treatment of genetic diseases, production of high yield and resistant varieties of food and crops, etc. Hence, we should utilize science and technology for the well-being of man and other living beings. We must use science and technology as a means to overcome the challenges faced by human beings. Science and technology are the products of human thought. We can justify this only if they are utilized for human benefit. That's all about this chapter. I really hope you have enjoyed the topic. See you soon with the knowledge on the biological world. Bye.
3 ലക്ഷത്തിലേറെ വരുന്ന ചോദ്യോത്തരങ്ങൾ ഏറ്റവും കുറഞ്ഞ ചിലവിൽ സ്വന്തമാക്കുവാൻ ഉടൻ തന്നെ ഡൗൺലോഡ് ചെയ്യൂ എൻക്ലാപ്സ് ദ ലേണിംഗ് ആപ്പ്.